This conference will now be recorded.
Hello and good evening to all of you. Hope you all are fine and enjoying the classes. So let's begin with the first lecture. Yes, good evening, Millie. Good evening, good evening, good evening, Jancy. Good evening to all of you. So this is my first lecture for CSIR NET. This is my actually the second lecture. The first lecture was for the strategy. So those who have not seen, they can watch it on YouTube. And today what I thought that I'll begin with your third unit, that is cell communication and signaling. So both these topics are different, okay? Cell communication and cell signaling. All of you can see the slides, right? Good evening, Priyanka. Good evening. Sorry, Priyamma. Sorry. Okay. So these are two different things, but they are always hold together, cell communication and cell signaling, right? What is communication? Communication is talking. And there are, for human beings, we have different ways of communicating. We can verbally, we can communicate, in verbally we can communicate. There are sign languages, through phones, through internet, through Wi-Fi, through Twitter, Facebook, and hell lot of things, right? So there are so many modes of communication. And what is the actual finale of this communication is that we are passing our messages, we are transferring our feelings, we are transferring our thoughts. That is the kind of signaling. Okay, that is the kind of signaling. Now any of the topics, whether it is your biology, whether it is your physics or chemistry, whatever it is, you must always relate it with your day-to-day life. When you are doing that, it becomes very easy for you to get the concepts easily, and very very lightly you can get the things very easily. Fine. So here what I have shown is a cell signaling, and there are two cells, and one cell is telling that, hi, can you hear me, endoplasmic reticulum, ER. Okay, and is there a way we can signal and talk? So the ER is replying, hey Golgi, yes, I can hear you, and we can signal each other now. So today's my topic is how we are going to signal the cells, and how these cells can transmit their information from one cell to another. Fine, understood everyone? What is the topic for today? It is cell signaling. Now see, here I have put a signal of a traffic. What does this light shows? What does this show? This figure. Can anyone reply what this figure depicts in the comment section? What does this figure depicts? Stop. Yes, right. It is stop. So this red color is a kind of a signal for us. And when this signal is depicted, we are ought to stop. Fine. And here, if it was a red green signal, there it would have shown that we should move. Fine. So these are the little bit signals which tells us without any communication. Does this is communicating? Does this is talking with us? No, it is not. But just through the signals, it is delivering its message that what we have to do. The same thing is done by ourselves. They do not talk with each other verbally, but through some signs and signals, they will pass their messages and they will communicate with each other. Fine. Now, the next thing. So what is this signal? Signal tells us when to stop, right? Just a second. Signals tells us when to start and when to stop. It can be used in different sign languages, which help us to understand very easily different types of gestures and symbols without talking verbally to each other. This is the thing which our cells do. Whenever you see this word cell signaling, just remember this traffic signal. Red means stop, and this it is doing without any verbal communication. It is just showing this red light, and we know what we have to do. Okay. Now from here, can anyone tell me what this is showing? Just turn your mind, just be creative. Don't tell me that this is a railway line, okay? But just tell me what this figure is depicting. Mixed signals. Very good, Charmi. Yes, mixed signals. Then cross connection. Very good, Jansi. Cross connection. Then group of different signals. Yes, yes. Cross connection. Yes. Okay, many of you are getting the things. So I think first slide से ही समझ में आने लगा. Very intelligent, huh? Yes, cross connection. Yes, Millie, right. Cross connection. So okay, yes. So see, first is long networks. These lines are very long. We cannot see where it is stopping and where it is starting. So the first thing is it is long networks. Second is, which everyone has told, cross linking. Cross linking. And the third thing is it is continuous. It is not breaking anywhere. It is continuous. So in our cells also, all these three things are shown. They are long networks of the cell, which is called as a cascade. I will show you in the next slides, but here only I'm telling the term. It is called as a cascade. Fine. Cross-linking means in our cell, nothing is like individual. Nothing is individual. One cell will communicate with other through many molecules, and those molecules will again communicate with thousands of the cells. So it is cross-linked. Nothing is like unique or single pathway. It is nothing like that. Everything is cross-linked. We will see the examples and the figures also. And it is continuous. Continuous means it is not like that that the signal is passing and then it takes a halt of 10 minutes and then it is again passing. No. If there is a cell signaling started, it will only end after the response is over. Only after that it will take rest. Okay. Otherwise, there are no breaks. It is ambiguous. It will just go in a fraction of second, the signal will be over. So three main characteristics of a cell signaling are these three things. And I have put these figures so that you can easily remember these things. Fine. Okay. Now, can anyone tell me which is this sport? Which is this sport? Everyone must have played in their childhood or must be playing now. What is this game? Relay race. Yes, very good, Deepshika. It is a relay race. What is the relay race? Yes, yes, Darshana. It is a relay race. Relay, right. What is a relay? Passing? Yes, it is passing, right, Deepshika? So what is being passed here? The stick, right? Whenever the one who is carrying the stick, passing a stick to another person continuously, yes. So whenever there is a stick in your hand, you have to run, right? It is that you have to run. Till that time, you are not passing that stick to another person, you cannot stop. You have to run. The same thing is your cell signaling. These sticks, you can consider it as a message. It is a message, okay, which these and all these characters which are here, they are the signal molecules. All of these, A molecule, B molecule, C, D, we will see what are these molecules, but just for now, just आप ऐसे remember करो कि ऐसे different molecules present होते हैं. सो ये हर एक मॉलिक्यूल के पास वो मैसेज है, एंड वो मैसेज वो लोग रिले रेस के थ्रू कंटिन्यूअसली पास करते जाते हैं जब तक कि एंड वाला पर्सन उस मैसेज को उसके टारगेट तक नहीं लेके पहुंच जाता. द रेस विल कंटिन्यू टिल the person is going at his target point. The target point may be, and mostly in many of the times it is the nucleus. It is the nucleus. Mostly the target is the nucleus. Fine. Mostly, yeah, not always, but mostly it is the nucleus. So your cell signaling is a type of a relay race. Now see the wordings what I have written. The branch molecular network, branch because it is cross-linked. I have shown it in the previous slide that it is a cross-linked network of activation and deactivation. It may be both of the stuffs. We will see it in our structured classes why it is both, but for presently you understood it as both. Okay, वो activate भी कर सकता है और deactivate भी कर सकता है. और ऐसे आपके नेट में क्वेश्चन आता है कि whether all the signaling molecules are activatory in nature, so the answer will be no. No, ठीक है. All the signaling molecules are not always activatory. कुछ signaling molecules होते हैं जो आपके एक्टिवेशन को रोकते भी है, वो डीएक्टिवेट करने के लिए भी काम करते हैं. सो सिग्नलिंग मॉलिक्यूल्स जरूरी नहीं है कि वो हमेशा एक्टिवेट ही करें, वो डीएक्टिवेट भी कर सकते हैं. तो आपका जो सेल सिग्नलिंग होता है दैट इज बोथ, इट इज एक्टिवेटिंग ऑल्सो एंड इट इज डीएक्टिवेटिंग ऑल्सो. द सिग्नलिंग मॉलिक्यूल्स विच आर लिंकिंग द रिसेप्टर एक्टिवेशन, I'll just tell you what is this receptor activation and everything. Just आप एक बार definition को देख लो. Receptor activation to the intracellular targets. Intracellular targets आपका कुछ भी हो सकता है. That is referred to as a signal transduction pathway or a cascade. Cascade इसीलिए क्योंकि it is a relay kind of thing. After one, next. The next will pass it on to the next, the next. Like this, your message will be flowing inside the cell. It will be running inside the cell. And that is why it is called as a relay kind of network. And why it is called a cascade? The cascade means one will activate the second, second will activate the third will activate the fourth. So it is like a cascade. 1, 2, 3, 4, 5, 6 till the target is reached. Just like this relay race. So whenever you are hearing this term cascade, you should remember this relay race. कि ओके द रिले रेस, जैसे हम रिले में स्टिक को ट्रांसफर करते हैं, यहाँ पे हम क्या ट्रांसफर करते हैं, मैसेजेस को हम ट्रांसफर करते हैं. वी आर ट्रांसफरिंग द मैसेजेस. फाइन. ओके, नाउ सी this is the plasma membrane. Okay, this is your plasma membrane. This is your plasma membrane. Okay, and this is one cell and this is another cell. Okay, and this cell is, it is very new. It is a newborn. It is first time it is going to activate. It is first time it is trying to go to its work. So he is very scared and he is telling, when I am around the membrane, how will I recognize and bind to her? I am very scared. Means that इसको पता नहीं है कि इसको कैसे अपना काम करना है. He doesn't know how to work. Okay, he doesn't know how to work. And what is his mother telling? No worries, son. Everything will be fine. You will be recognized. Right? Now his mother is telling कि आप कुछ भी चिंता मत करो, आपका सारा काम जो है वो हो जाएगा. So this is the molecule. This is the signal molecule, and this signal molecule is ought to give the signal transduction, and he has to, और ये कब signal transduction देगा, when he will be giving the signal transduction. दो methods होते हैं, या तो ये cell membrane पे bind करे और या तो फिर ये cell membrane के अंदर जाके bind करे. Binding is the word. जब तक कि बाइंडिंग नहीं होता है तब तक कुछ भी नहीं होता है. इट हैज टू बी बाइंडेड. बाइंड होना बहुत जरूरी है. तो ये जो है आपका जो मॉलिक्यूल है, सिग्नल मॉलिक्यूल, ये क्या करेगा, ये बाइंड करेगा. या तो फिर ये मेम्ब्रेन पे बाइंड करेगा यहाँ पर, या तो फिर ये इनसाइड दिस यहाँ पे ये बाइंड करेगा अंदर की तरफ. एक सेकेंड, या तो फिर ये यहाँ पे बाइंड करेगा, यहाँ पर सपोज की कोई मॉलिक्यूल है तो वो यहाँ पे जाके बाइंड करेगा। तो इट हैज टू थिंग्स, या तो वो मेम्ब्रेन पे बाइंड करेगा या तो फिर वो इनसाइड द मेम्ब्रेन ये बाइंड करेगा। बट अभी इसको डर लग रहा है, ही इज स्कैर्ड कि ये कैसे करेगा, यहाँ पे तो न जाने कितने सारे चीजें होंगी, ही इज वेरी न्यू, देयर विल बी लाइक यहाँ पे एक मॉलिक्यूल होगा, यहाँ पे एक होगा, यहाँ पे एक होगा, यहाँ पे एक होगा, देयर विल बी सो मेनी मॉलिक्यूल्स हियर, सो मेनी मॉलिक्यूल्स। So he is afraid that ये कैसे इसको पता चलेगा कि which he has to go and bind. So इसकी जो मम्मी है, she is telling don't worry, you will be recognized. So ये जो इसकी मम्मी बोली कि आप चिंता मत करो, आपको जो ये मेम्ब्रेन पे जो आपका जो चीज होगा वो पहचान लेगा। फाइन, सो लेट्स सी वो कैसे होता है। Everyone is understanding? Any doubts? Okay, yes. Okay, now see, here is the thing. This is your plasma membrane. This is your plasma membrane. Fine. I'll take a highlighter. This is your plasma membrane, double layer plasma membrane. And this is called as a transmembrane receptor. This is called as a receptor. Fine. This is called as a receptor. And this is its active site. This is called as its active site. Active site वो चीज होता है जहां पर आपका सिग्नल मॉलिक्यूल आके बाइंड करेगा. That is called as an active site. Just like as your enzyme, where your enzyme में जहाँ पे आपका active site पे आपके ligand bind होता है, the same way here also, because these are the protein molecules. All these are protein molecules, right? So this is called as a this is called as a receptor, which is a transmembrane receptor. Transmembrane का मतलब होता है that it can traverse through this membrane. It can traverse through this membrane. जब ये पूरे के पूरे membrane को cross करता है like this, it is called as a transmembrane receptor. And ये जो इसके top में present है, that is called as an active site of the receptor. That is called as an active site of the receptor. Understood? Now see what will happen. Now, these are the different types of signal molecules. These are different types of signal molecules. This is A molecule, this is B, and this is C molecule. Now, this molecule is not knowing that where I have to bind. I don't know where I have to bind. But can you see this active site? This active site has a specific type of a shape. Right, this active site has a specific type of shape. Now what will happen is that this active site will be able to bind only with that kind of molecule, only with that kind of signal molecule, which shape is, whose shape is ekdam consequent with that shape. It should bind, it should come and lock with this one. This cannot bind, this cannot bind, only this can bind to this receptor. So it is very important that you must have you must have listened about the antigen antibody interactions, right? Then enzyme ligand interaction. The same thing is happening here also. Every receptor is unique to a signal molecule. And what is that uniqueness? That uniqueness is called as the active site, and on that active site your signal molecule will come and join. And what difference can you see between these two figures? One is the signal molecule has joined. What is the other difference which you can see? Comment. What is the other difference which you can see? See the figure. Pathway. Okay, next. Difference in receptor. Okay, Vidushi. Next. See the figure, just you have to see the figure and then you have to tell me. See the figure and then tell me. Receptor changes. Very good, Jancy. Yes, receptor changes. Okay, the word is called as one way. Okay, the word is called as confirmation change. The word is called as confirmation change. Can you see here and can you see here? Before the molecule has joined and after the molecule has joined. यहाँ पर आपका molecule ने join नहीं किया हुआ, ये जो आपका signal molecule है उसने join नहीं किया है, but यहाँ पर आपके signal molecule ने join किया है. And जैसे ही the signal molecule attaches with your receptor molecule, the confirmation of this transmembrane receptor changes. Confirmation का मतलब होता है उसका जो shape है, the shape has changed. The shape has changed. I have shown here a very drastic change, but ऐसा नहीं होता कि it is so drastic change, but just to depict that yes, there is a difference in the configuration. So that is why I have changed the shape. So the thing is that whenever there is attachment of the ligand molecule onto your active site, always your receptor conformation will change. And as soon as this conformation changes, this is a trigger. ये trigger रहता है, cells को ये एक message pass करता है कि now you have to start working. राइट ना, यहाँ पे कोई भी वर्बल कम्युनिकेशन नहीं हो रहा. आपको याद है वो ट्रैफिक सिग्नल रेड मतलब कि यू हैव टू स्टॉप. जस्ट लाइक दिस, यहाँ पे जैसे ही कंफर्मेशन चेंज हो जाएगा, एज सून एज देयर इज कंफर्मेशन चेंज हियर, द सेल्स गेट द सिग्नलिंग दैट यस आई हैव टू स्टार्ट माय वर्क. And this can be through various responses. Okay, this can be through various responses. The first response can be as soon as this molecule is binding to this active site, there may be joining of certain things which is called as the secondary molecules, which is called, these are the phosphorylation sites. No, sorry, these are not secondary molecules, these are called as the kinases. These are called as the kinases. That kinases are the things which are very important for phosphorylation. I have written the word here. It is called as phosphorylation. Phosphorylation means these kinases will add the phosphorus groups. This kinases will add the phosphorus group. So can you see, it is kind of relay race. First, this will come and join. Second, this will change its shape. Third, this phosphorylation will occur. And as soon as this, now this is one type that phosphorylation can occur. And the second type is that I have shown here the arrow. Okay. Now this arrow depicts that the gate is open. This is like a gate. As soon as till when the receptor is not bound, this gate is closed. ये जो gate है आपका, ये जो gate है ये बंद रहता है. पर जैसे ही आपका ये receptor आके bind करेगा, ये जो gate है आपका, ये gate open हो जाएगा. किस चीज के लिए open हो जाएगा? For various things like the ions, the gases, the hormones, like that. Mostly for ions, the gate is open. The gate is open for the ions, and as soon as the ions come inside the cell, the signal transduction will start. Or this is phosphorylation. So there are two ways by which the signal transduction can pass. First is through phosphorylation, and second is through gated channels. There are two things. First is through these kinase molecules, and the second is through the gated channels. Gated channels को आप ऐसे समझ सकते हैं कि वो एक दरवाजा है. इट इज लाइक अ गेट. एंड वो कब ओपन होगा? जब आप उसके अंदर की लगाओगे. की क्या है? आपका ये सिग्नल मॉलिक्यूल है. तो जैसे ये सिग्नल मॉलिक्यूल यहाँ पे आके ज्वाइन करेगा, ये जो गेट है ये ओपन हो जाएगा, एंड यहाँ से पूरे के पूरे आयंस जो है वो पास होने लगेंगे. सो देर आर टू वेज. वन इज फॉस्फोराइलेशन एंड द अदर वन इज योर गेटेड चैनल. सो दीज आर द टू थिंग्स. Now, what will be the next thing? What the other molecules will do? They will activate and amplify. We will see what is amplification in the next slide. And here what comes a very important molecule which is called as the second messenger, like the CAMP, the second messenger. Now, why it is called as a second messenger? What is the first here? What is the first thing here? I have written here second. What is the first one? Can anyone comment? Recognition. Okay. Okay, recognition. Then first molecule which was bind. Yes, right. The first is this one. This one is the first. The first is your signal molecule which has joined. अगर ये join नहीं होगा तो आपका नीचे का कोई भी process, yes, recognition by the active site, right, Manasvi? This is your first one, because if this is not happening, all these things is not going to happen. So this is your first one. When your signal molecules is joining here, this is your first one. And after this thing, whatever is the cascade going on, the cascade is then giving you the second messenger. That is why this word is second. This comes in your net exam. Then why it is called a second messenger? Because the first one is the initial one, the signal molecule. That is called as a first messenger, and this is your second messenger. And yes, what? Sorry, any problem? Hello.
Yes, ma'am, continue.
Okay. So this is your second messenger, and what this will do, this will help in the amplification of your cascade. Now amplification is very important, क्योंकि जितना आपका signal strong होगा उतना ज्यादा आपका expression अच्छा होगा. If it is a very weak signal, then it is not going to give you the response. So that is why amplification is very important. Amplification means one signal is to be multiplied into 1,000 times. Amplify means like PCR. PCR mein you are giving few genes, like you are giving 10 genes, and 10 genes after PCR it is converting into like 10,000 genes. So that is called as amplification. So here also the same thing happened. Only one receptor, only one signal, but till here it is like thousands of the molecules are present inside the cell, thousands of molecules, and these 1,000 molecules will activate the transcription factor. You should know this term. It is called as the transcription factor. It is called as the transcription factor. Okay, this is called as the transcription factor. And what this transcription factor will do, this transcription factor will go inside your nucleus and it will help in the expression of the genes. आपके ट्रांसक्रिप्शन फैक्टर का सिर्फ और सिर्फ नाम से पता चलता है. ट्रांसक्रिप्शन फैक्टर मतलब कि वो ट्रांसक्रिप्शन में हेल्प करेगा. और ट्रांसक्रिप्शन मतलब क्या होता है? जब डीएनए से आपका एमआरएनए बनता है, जब डीएनए से आपका एमआरएनए बनेगा उसको हम कहते हैं ट्रांसक्रिप्शन. और जो फैक्टर वो करने में हेल्प करता है दैट इज कॉल्ड एज अ ट्रांसक्रिप्शन फैक्टर. Okay, so this second messenger, उसका example है CAMP, cyclic adenylyl monophosphate. आप बस CAMP याद रखो. CAMP, it will activate this transcription factor, and this transcription factor will go inside the your nucleus. Will go inside your nucleus, and nucleus में जाके ये क्या करेगा? आपके genes को express करेगा, and ये आपका final target था. न्यूक्लियस तक पहुंचना इसका फाइनल टारगेट पॉइंट था. फाइन. तो कहां से शुरू हुआ था आपका जो सिग्नल ट्रांसडक्शन था? मेम्ब्रेन से शुरू हुआ था. एंड वो कहां पे जाके एंड होता है? इन द न्यूक्लियस में जाके वो एंड होता है. एंड आप देखेंगे कि धीरे धीरे the signal is transferring. It is like a cascade. It is like a relay race. And यहां पर आके आपका एम्प्लीफिकेशन हो जाता है. देयर इज इंक्रीज इन द सिग्नल स्ट्रेंथ. एम्प्लीफिकेशन हमेशा याद रखना है, इंक्रीज. The number is multiplying 1,000 times. It is multiplying, and that is activating this transcription factor, and then it is expressing your genes. Understood, everyone? Any doubt till here? Okay, fine. Okay, now see where these cell signaling occurs. What are the examples of this cell signaling? The first example is when your cells are dividing. If the cell wants to divide, it has to be given a signal. The signal may be a growth hormone. The signal may be a growth hormone or some kind of dividing factors, right? Some kind of dividing factors, and that dividing factors will tell this cell that come on, now you have to divide. So this is one example where the cell signaling will occur. The second is your apoptosis. Apoptosis is programmed cell death. That means the cell is undergoing a suicidal pattern where it is killing itself. Okay, and that it is doing for it is like a सर्विस अ नेशनल सर्विस जैसे कि हमारे आर्मी एंड जो हमारे डिफेंस के पर्सन्स करते दे आर sacrificing their life for our country. The same thing is done by these cells. They are killing themselves for the benefit of the other cells. Okay, this may be the case whenever there is a virus infection. Suppose this cell is infected by a virus. So what, and since this virus is contagious, it can be transferred to other cells which are nearby. So what this cell will do? This cell will kill himself, so that this virus cannot move and go to the other cells. Okay, so this is kind of a sacrifice this cell is doing, which is called as apoptosis or programmed cell death. So here also it is very necessary that there should be a signal. There is a death signal. Okay, there is a death signal which is given by the cells, so that these cells now know that okay, I have to suicide. Fine. So here also cell signaling gives a very important work. The next one is your movement inside the cell. When the cells have to transfer the things from one organelle to other organelle. Like the first slide where I have shown you कि जो एंडोप्लाज्मिक रेटिकुलम है वो गोल्गाई से बात कर रहा है कि क्या हम एक दूसरे के साथ सिग्नलिंग ट्रांसफर कर सकते हैं. द आंसर इज यस. एंड व्हाट वाज द आंसर? दिस microtubule based movement. The answer is microtubule based movement. Okay, these are the microtubules, and these microtubules are like roads. These are the roads. Okay, and ये roads क्या करता है? ये roads के ऊपर ये molecules है which are called as the motor proteins. Dynein, kinesin, all these are motor proteins. Okay, like टैक्सीज. ओके, ये आपका रोड है और इसके ऊपर ये ट्रेन, टैक्सी, कुछ भी आप समझ लो. ये टैक्सी है जो कि मूव कर रहा है, और ये क्या लेकर मूव कर रहा है? आपका ऑर्गेनल्स. ऑर्गेनल्स में कुछ भी चीजें हो सकती है, एनी टाइप ऑफ कार्गो. उसको अगर एंडोप्लाज्मिक रेटिकुलम से गोल्गाई में ट्रांसफर करना है, गोल्गाई से उसको अगर न्यूक्लियस में ट्रांसफर करना है, न्यूक्लियस से अगर उसको सेल मेम्ब्रेन पे ट्रांसफर करना है, wherever it wants to deliver the things, it is like a pack and parcel thing. So these are the microtubule based networks, and इसको वर्क करने के लिए भी it is very important कि आपका सेल सिग्नलिंग बहुत स्ट्रॉन्ग होना चाहिए. जब तक सेल सिग्नलिंग स्ट्रॉन्ग नहीं होगा, these molecules, these motor proteins, they will never come to know that which path they have to follow. इनके पास आईज थोड़ी ना होती है. They cannot see. They can only sense. They can only sense the signals. So only those signals can be sensed, and they will transfer the stuffs to the correct and targeted organs. Understood? So this is your third example where the cell signaling occurs. Then your next example is, and very important example is, your synaptic transmission. A very, very, very important example. And this neurotransmitter things, it always comes in your net exam, in your 4 marks, in your 2 marks. It is a definite question. Okay, so this is also kind of relay system where the molecules are transferring from this nerve to this nerve. Now this nerve is called as the presynaptic nerve. Pre means pehla. Okay, this is the presynaptic nerve, and this is your postsynaptic nerve. Postsynaptic means बाद में. तो आपका जो नर्वस सिस्टम में जो ट्रांसमिशन होता है, that is also an example of a cell signaling pathway where these are the signaling molecules which are released. These are the signaling molecules which are released. You can see here it is released here, and then they will go and bind to this receptors molecules which are present on the postsynaptic nerve के membrane पे present है. ये सारा जो आपको दिख रहा है, these are called as the receptor molecules, and these molecules, the signal molecules will go and join here, and then your cascade will start here. And mostly here these are like channels, gated channels. मैंने आपको 2 टाइप बताए थे, एक था फॉस्फोराइलेशन और दूसरा था गेटेड चैनल्स. तो ये जो है ये आपका गेटेड चैनल्स है. आपका जो नर्वस सिस्टम में जो जिस टाइप का जो सेल सिग्नलिंग वर्क करता है वो है गेटेड चैनल्स. गेटेड चैनल्स है जो कि आपका सिनेप्टिक ट्रांसमिशन में वर्क करता है. दिस इज अ डेफिनेट क्वेश्चन व्हिच इज एवरी टाइम आस्क्ड इन योर एग्जाम्स. एंड मोस्टली व्हाट आर दोस गेटेड चैनल्स? दे आर मोस्टली ion gated channels or ligand gated channels, which we will study in our structured classes. यहाँ पे आपको मैं सिर्फ introduce कर रही हूँ कि okay, these are the gated channels which are present in these neurotransmission, which is very important, and this is a very basic example of your cell signaling mechanism. समझ में आया आपको चारों एग्जांपल्स? कहां कहां पे आपका सिग्नल मॉलिक्यूल्स वर्क करता है? सेल ग्रोथ, सेल डिवीजन, एपोप्टोसिस, मूवमेंट एंड योर सिग्नल ट्रांसमिशन, सिनेप्टिक ट्रांसमिशन और सिग्नल ट्रांसमिशन. Okay, now okay Vidushi. Okay, now see, this is one of your net question which was asked, that arrange these sequence in the correct order of the cell signaling pathway. What is the correct order of the cell signaling pathway? So what is the first thing called? The first thing is called as the reception. Reception means your signal molecule will be received, that is synthesis and release of the signaling molecules by the signaling cell, and transport of the signaling to the target cell. Okay, अगर वो टारगेट सेल तक नहीं पहुंचेगा तो ना तो आपका सिग्नल मॉलिक्यूल अटैच होगा टारगेट सेल पे और ना तो आपका आगे का प्रोसीजर फॉलो होगा. सो इट इज वेरी इंपॉर्टेंट दैट द सिग्नल मॉलिक्यूल हैज टू बी transferred to the target cell. So it will come and attach to your target cell. And these two processes are called as reception. This is called as reception. So the first thing is there is production of the molecule and it is going and attaching to the target cell. This is the first procedure. First and second procedure, right? Then it is called as amplification. This is your first one. This is your second one, and this is your third one. Amplification means the cascade, the relay system, the networking inside the cell, the signaling which is going inside the cell, that is called as amplification. Once the target has come and attached to its specific point, it will start amplifying the signal. And after the amplification, there will be the initiation of the signal transduction pathway, which is called as the response. That is the inside molecule. The secondary messengers will be formed, or there will be phosphorylation of the transcription factor. The transcription factor will go inside the nucleus. Whatever I have discussed in the previous slide, जो भी अभी मैंने discuss किया, तो वो जितनी भी एक बार बाइंडिंग के बाद जो भी नीचे का जो प्रोसेस होता है, दैट इज कॉल्ड एज अ रेस्पॉन्स. और वो किसके रेस्पॉन्स में हुआ है? आपका ये रिसेप्शन के रेस्पॉन्स में हुआ है. ओके, दिस इज द रिसेप्शन. जो भी मॉलिक्यूल, जो भी आपका सिग्नल मॉलिक्यूल आकर अटैच हुआ है आपके टारगेट सेल से, वो आपको ये चीज करने के लिए हेल्प कर रहा है, रेस्पॉन्स करने के लिए. एवरी एक्शन हैज एन इक्वल एंड ऑपोजिट रिएक्शन, राइट. वो जो आपका थर्ड लॉ है वो यहाँ पे काम करता है. जो भी चीज आपका टारगेट है यहाँ पे, ये टारगेट सेल आपका रिस्पॉन्स करने में हेल्प करेगा आपके सेल के अंदर. सो दिस इज योर सीक्वेंस, दिस इज योर सीक्वेंस ऑफ द सेल सिग्नलिंग. आप और ये बहुत इंपॉर्टेंट क्वेश्चन है, आपको सीक्वेंस पता होना चाहिए कि सबसे फर्स्ट, मैंने आपको फिगर में भी दिखा दिया और मैं यहाँ पे भी आपको बता रही थी, फर्स्ट आपका सिग्नलिंग मॉलिक्यूल बनेगा, वो आके अटैच होगा, फिर वो जैसे ही वो बाइंड होगा नीचे का जो कंफर्मेशन चेंजेस होगा, एंड देन एम्प्लीफिकेशन शुरू होगा, एंड देन जो भी रिस्पॉन्स है दैट रिस्पॉन्स विल बी गिवन. सो दिस इज योर सीक्वेंस. Now this topic is very important for your third part of your CSIR net. Second part में से तो definite questions आते ही हैं, but third part which is a analytical part, all these things analysis वाला जो portion रहता है उसमें से आपको कम से कम नहीं तो 6 to 7 questions आपको यहाँ से मिल जाएंगे signaling से, signaling and communication, because all these are research analytical types of questions. ओके, नाउ सी, यहाँ पे आपको मैंने एक एक्चुअल पाथवे बताया हुआ है. इन जनरल क्या क्या चीजें होती हैं, वी विल सी व्हाट आर दीज टर्म्स इन शॉर्ट, वेरी शॉर्ट. आपको बस पता होना चाहिए कि ये क्या चीज होती है, एंड देन वी विल सॉल्व सम क्वेश्चंस व्हिच हैज कम इनटू योर नेट एग्जाम्स. वी विल सॉल्व दोस क्वेश्चंस. ओके, नाउ सी, this is your receptor molecule, this is your receptor molecule, and what is this? This is your signaling molecule. So this signal has come and joined to this receptor molecule, and as soon as it has joined, there is something called as the scaffold protein. Okay, there is a scaffold protein. It is like a scaffold, मतलब कि helping protein होता है. Okay, ये helping protein किसके लिए help करेगा? Helping to bind these proteins. You can see the grooves here. इसको grooves कहते हैं. These are called as the grooves. And ये grooves में these protein will come and attach. Okay, these are the grooves. And ये इसको कैसे पता चलेगा? How these proteins will know that we have to come and attach? Because as soon as these molecules join here, there is a conformation change occurring here. This all these grooves will be open up. पहले ये सब बंद था क्योंकि कोई कंफर्मेशन चेंज नहीं हुआ था, क्योंकि ये मॉलिक्यूल यहाँ पे अटैच नहीं था. जैसे ही ये मॉलिक्यूल यहाँ पे आके अटैच करेगा, वैसे ही ये जो गैप्स है ये सब ओपन हो जाएंगे, और ये गैप्स में आके आपका ये नीचे का जो कैस्केड मॉलिक्यूल्स है ये आके यहाँ पे ज्वाइन करेगा. अंडरस्टूड? आपको कैसे जो हमने पहले एक जनरल फॉर्म में किया था, ये उसका थोड़ा expanded version है. So these are the scaffold protein जो कि help proteins रहता है, and these are the relay protein. Once more, ma'am. Okay, I'll explain you once more. See, this is your plasma membrane. This is your plasma membrane. Okay, this is outside the cell. This is outside, and this is inside the cell. Okay, this is cytosol. Cytosol cell के अंदर होता है, तो ये cytoplasm है पूरा, और ये जो है ये आपका न्यूक्लियस है. फाइन. तो एक मैसेज मिला है कि यू हैव टू ग्रो. ओके, सो यहाँ पर एक सिग्नल मॉलिक्यूल आके यहाँ पे अटैच हुआ है. ओके, दिस इज योर सिग्नल मॉलिक्यूल, एंड ये सिग्नल मॉलिक्यूल कहाँ पे आके अटैच होगा? वहीं पे आके अटैच होगा जहां पर ये एक्टिव साइट उसको बाइंड करेगा. देयर आर thousands and thousands of receptors which are present on this plasma membrane. But this receptor will be recognized only by this receptor. Only by this receptor this is recognized. Why? Because इसके और इसके shape में एक matching है. That is why this molecule will come and attach here. Okay. And then what will happen? जैसे ही ये attach करेगा, there is a conformation change in this scaffold protein. Scaffold protein is a kind of helping protein. It is a kind of helping protein which will help to bind these other proteins. Okay, you can see here. Okay, it is like it is attaching this receptor molecule with all these molecules. It is kind of attaching. Scaffold protein is a kind of attaching the things here. So what are these relay proteins and adapter proteins? These are a kind of cascade proteins. These are a kind of cascade proteins. Okay. Cascade means they will transfer the signal to the next one and next one and next one. आपको बस ये नाम याद रखना है कि relay proteins होता है, adapter proteins होते हैं. Okay, now see here. Now from here adapter proteins you can see यहाँ पे एक molecule बना है. यहाँ पे suppose कि ये जो molecule यहाँ पे बना था, this one molecule can signal 3 pathways. This is one pathway, this is second pathway and this is third pathway. तो हमने जो starting में जो पढ़ा था cross connection, याद आया? Cross linking. यहां से वो चीज आपको क्लियर होगी कि आपका ये एक मॉलिक्यूल कैन कैस्केड अदर टाइप्स ऑफ सिग्नलिंग पाथवेज आल्सो. तो ये आपके नेट में क्वेश्चन आता है कि वेदर द सिग्नलिंग मॉलिक्यूल्स आर यूनिक और नॉट. ओके, so the answer is false. The answer is no. The signaling molecules are not at all unique. They can signal other pathways also. You can see here it can do this also, it can do this also, and it can do this also. Understood? So the signaling molecules are not at all unique. They are not at all unique. Now these are the amplification molecules. See here, these are the second messengers. And यहाँ पे आपका सिर्फ एक ही signal था, only one signal, but this one signal has amplified. It has amplified to so many signal molecules. It has amplified, right? And this amplification will go to this messenger proteins, and this messenger proteins will then, this messenger protein can be your transcription factor. Okay, जैसे मैंने आपको example दिया, ये कुछ भी हो सकता है. ये transcription factor है, suppose for example, and this transcription factor will go and bind to its target and then to DNA, and then the gene transcription will start and the response will be given. Understood, everyone? These are the technical terms which you have to remember. These are the technical terms. Bifurcation proteins means दो direction में इसने target कर दिया. Bifurcate means to divide. So it has divided it so that this molecule can go and initiate another pathway. This molecule can go and initiate another pathway. So that is why it is called as a bifurcation protein. So you should know these kind of technical terms in cell signaling pathway. Understood? Now clear, everyone? This is the basics. Actually, there are so many pathways which we have to learn and which are very important for your NET exam, but that we will do only in our structured classes. But here I'm giving you an overview, and you can see from this overview also, you can solve so many questions. You can solve so many questions. Okay, okay, everyone. Okay, fine. Let's go to the next one. See, these are the, sorry. See, now these are the examples of your membrane protein. These are the examples of your membrane proteins, and all these membrane proteins will give rise to one kind of response. Everyone has a different type of response which is given, and we have to study all these kinds of pathway. These are the important ones which I have mentioned here. Okay, these all we have to study. One thing you have to know here, this is called as a G protein coupled receptor, GPCR. This is called as a G protein coupled receptor, and this receptor is the largest one. You should know this. This protein is the largest one in size. Okay, it is a 7-membrane transmembrane protein. 7 transmembrane protein. We will see the structures when we are doing individually the GPCR, but you should know here that this GPCR is the largest one. Then you can see here, see this SMAD, this MAPK, all these are kind of signal molecules. These are messenger molecules. All these are messenger molecules. Examples है उसका ये. Okay, these are the kinases. These are kinases. K means these are kinases. Kinases मतलब वो proteins जो phosphorylate करेगा next protein को. ये फॉस्फोराइलेट करेगा. जहां पे भी आपको के दिख रहा है, ये इसको फॉस्फोराइलेट करेगा, ये इसको फॉस्फोराइलेट करेगा. ओके, तो दिस इज काइंड ऑफ कास्केड, और ये ट्रांसक्रिप्शन फैक्टर पे जाएगा जीन के ऊपर, और ये अपना डीएनए से एमआरएनए को ट्रांसक्राइब करने में शुरू कर देगा. ओके, सो दिस इज अ काइंड ऑफ रिले सिस्टम. दिख रहा है, ये इसको फॉस्फोराइलेट कर रहा है, ये इसको कर रहा है, ये इसको कर रहा है, ये इसको कर रहा है. सो दिस, अगर इसके अंदर कुछ प्रॉब्लम हो गया ना, अगर सपोज की इफ देयर इज एनी प्रॉब्लम इन दिस वन, if suppose any mutation occurs in this one, ये पूरा का पूरा पाथवे ब्लॉक हो जाएगा, ये काम ही नहीं करेगा आगे. दिस विल ब्लॉक. और ये आपका जो आपका सेट 3 का जो पेपर आता है, दैट इज पार्ट सी. जो पार्ट सी आता है उसके अंदर ऐसे क्वेश्चंस पूछते हैं, और ये बहुत इंपॉर्टेंट मैकेनिज्म है कैंसर को रोकने के लिए. दिस इज अ वेरी इंपॉर्टेंट मैकेनिज्म जो कि आपका कैंसर को ब्लॉक करता है. बिकॉज इन कैंसर कोई सा भी पाथवे अपने आप चालू हो जाता है. इट इज लाइक ऑटोनोमस. कोई सा भी पाथवे जो है, इट कैन स्टार्ट ऑन इट्स ओन. सो वी आर टारगेटिंग ऑल दीज प्रोटीन्स. कोई सा भी हम प्रोटीन्स को टारगेट कर सकते हैं, एंड वी कैन ब्लॉक दैट प्रोटीन. अगर ब्लॉक कर देंगे तो आगे का प्रोसीजर होगा ही नहीं. ऑल दीज प्रोसीजर विल बी ब्लॉक. सो अगर ये सारा प्रोसीजर नहीं होगा तो आपका ट्रांसक्रिप्शन फैक्टर यहां नहीं जाएगा. ट्रांसक्रिप्शन फैक्टर यहां नहीं जाएगा तो आपका जीन ट्रांसक्राइब नहीं होगा. जीन ट्रांसक्राइब नहीं होगा तो आपका जो सेल्स है वो डिवाइड नहीं होगा. सो दिस इज वेरी इंपॉर्टेंट, एंड यू शुड सी हियर वन थिंग कि यहाँ पे देखो क्रॉस कनेक्शन भी हो रहा है. ये प्रोटीन जो है, ये वाला जो प्रोटीन है, वो आपके इसको भी कर रहा है टारगेट और इसको भी कर रहा है टारगेट. सो दीज आर सम ऑफ द वेरी इंपॉर्टेंट एग्जाम्पल्स जो कि आपके सेल सिग्नलिंग में आपको पढ़ना है. और ये हर एक जो है, इट इज अ डिफरेंट टाइप ऑफ सेल सिग्नलिंग पाथवे. डिफरेंट टाइप ऑफ सेल सिग्नलिंग पाथवे, हर एक के इंपॉर्टेंट कैरेक्टरिस्टिक्स है विच वी हैव टू स्टडी. And one thing you should remember, ये जो है, this is the largest one. It is 7 membrane spanning domain, and it is the largest one. Understood? Okay. Now see, ये मैंने वैसे भी explain कर दिया, but one more time I'll just tell you. रिसेप्टर सबको समझ में आ गया? रिसेप्टर मतलब कि आपका जो मेम्ब्रेन में प्रेजेंट होता है, दैट इज कॉल्ड एज अ रिसेप्टर, जो कि रिसीव करेगा आपके सिग्नल को. मैम यू आर नॉट ऑडिबल. हेलो, एम आई ऑडिबल? हेलो, हेलो.
Yes, ma'am, you're audible. Dear Millie, check your connection issue.
Okay, okay, okay. हाँ, ठीक है, ठीक है. So this is your receptor. So the receptor is the one जहां पे रिसीव होता है आपका सिग्नल. सो दिस इज योर रिसेप्टर. और ये क्या है आपका सिग्नल मॉलिक्यूल. दिस इज योर सिग्नल मॉलिक्यूल. नाउ डेलिबरेटली मैंने आपको ये वर्ड नहीं बताया था लाइगैंड, बिकॉज मुझे आपको यहाँ पे शो करना था. आपके सिग्नल मॉलिक्यूल को हम लाइगैंड मॉलिक्यूल भी बोलते हैं. ये जो आपका सिग्नल मॉलिक्यूल है इसको हम लाइगैंड मॉलिक्यूल भी बोलते हैं. और बहुत सारे टाइप्स के लाइगैंड मॉलिक्यूल्स होते हैं. देयर आर n number of types of ligand molecule which can activate n number types of the receptors. Okay, then there are two types of the signaling. One is your intercellular signaling and one is your intracellular signaling. इंटरसेल्युलर मतलब क्या होता है, इसमें हमेशा कंफ्यूजन होता है लोगों को. इंटर मतलब हमेशा याद रखना एक सेल के साथ दूसरा सेल. इंटर इंटर मतलब एक सेल के साथ दूसरा सेल. और जहां पर भी वर्ड ए आ जाता है मतलब अपने आप में. ए मतलब अपने आप, यानी कि एक सेल अपने आप ही कम्युनिकेट करेगा उसी के साथ, अपने आप ही कम्युनिकेट करेगा. एंड इंटरसेल्युलर मतलब कि वन सेल विल कम्युनिकेट विद अदर सेल. एंड इंट्रासेल्युलर मींस वन सेल विल कम्युनिकेट विद herself or himself or itself, जो भी आप जेंडर लगाना चाहते हो. So it is intracellular. And this question is also asked in your net exam कि intracellular क्या होता है, intercellular क्या होता है, and we get confused because उस समय याद नहीं आता. तो ऐसे कुछ आपको mnemonics याद रखने, जैसे कि A अगर है तो मतलब it is अपने आप में, by itself it will do. Okay, now when it is doing this intercellular signaling, that is when one cell is communicating with another cell, one thing matters which is called as the distance. It is very important. क्योंकि suppose for example if I have to go to a nearby place, okay, मेरे घर से सिर्फ 1 किलोमीटर की दूरी पे, I have choices. मैं चल के भी जा सकती हूँ, मैं बाइक से भी जा सकती हूँ. Okay, but if I have to go to a far off places, okay, so I have to take a bus. अगर मुझे दूसरे स्टेट में जाना है, then I can take a railway or I can take a flight. Okay, so these are the different things, and the distance will decide that what mode we are going to take, right? So here also, when there is a cell to cell communication, the distance matter, and the distance will decide what type of cell signaling is going to occur. And this is very important, very important. Okay, what kind of cell signaling is going to happen? The first is your contact dependence. The contact dependence is also called as juxtacrine signaling. Contact dependent means, see, this is one cell, this is second cell. They are very close to one another, very close. And these are the gates. ये gates हैं. ये gates कुछ भी हो सकता है. Like अगर animal है तो इसके अंदर gap junctions present होगा. Gap मतलब कि like doors. Okay, दरवाजा है. अगर मुझे अपने neighbor को कुछ देना है तो मैं अपना दरवाजा खोल के I can pass it on. So this is one cell, this is second cell, and they are very close to one another. But how they can communicate? They can communicate through these gaps. If it is animal, it is called as gap junction. And if it is plant, it is called as plasmodesmata. It is called as plasmodesmata. So through here, the signals can pass. Understood? This is the signaling molecule, and this is passing the signal to this molecule through this gap junction. And this is called as juxtacrine signaling. The question comes, what is juxtacrine signaling? Mostly the contact dependent is not asked, but it is called as juxtacrine signaling. Understood? Now everyone is clear what is juxtacrine signaling? When the cells are very close enough, but only they can communicate through these gates, then it is called as juxtacrine signaling. Here the condition is the cells have to be very close, very close. You can see here the cells are very close, कोई भी gap present नहीं है. So this is called as your juxtacrine signaling. The next one is paracrine signaling. The next is your paracrine signaling. Para means, you can see here there is a gap. There is a gap. The cells are not very close together, but they are in neighbors. They are in neighbors. It is like ये एक बिल्डिंग में रहता है और ये एक दूसरे बिल्डिंग में रहता है. They are not. They are kind of neighbors, locally surrounded है. This is in the local signaling. Okay, local signaling. So here also the molecules are transferring, and this kind of transfer, this kind of signaling is called as paracrine signaling. The molecules can diffuse between them. Okay, here the ligands are very easily reabsorbed and very easily degraded. क्यों ये जल्दी reabsorb होते हैं, क्यों ये degrade होता है? क्योंकि अगर ये already present रहेगा ना यहाँ पे बहुत समय तक, तो ये continuously क्या करेगा? अगर ये बहुत सारे समय तक अगर यहाँ पे प्रेजेंट है, तो ये क्या करेगा कि इसको कंटिन्यूअसली ये इसको टारगेट करता रहेगा, कंटिन्यूअसली टारगेट करता रहेगा. और अगर ऐसा हुआ तो ये इसके ऊपर एक म्यूटेशन का इफेक्ट आ जाएगा. दैट इज वाई ये बहुत जल्दी एब्जॉर्ब भी हो जाता है और एक बार अपना काम करने के बाद ये इस पूरे को डिग्रेड भी कर देते हैं. दिस इज वेरी इंपॉर्टेंट कि ये रीएब्जॉर्ब भी होता है जल्दी एंड इट इज डिग्रेडेड ऑल्सो वेरी इजिली. एंड दिस इज कॉल्ड एज अ लोकल सिग्नलिंग. अ लोकल सिग्नलिंग. इसका एग्जाम्पल है आपका सिनेप्टिक सिग्नलिंग, जो मैंने आपको दो नर्वस के बीच में बताया था, याद है, प्री सिनेप्टिक एंड पोस्ट सिनेप्टिक. तो ये जो सिनेप्टिक सिग्नलिंग है ये इसका बेस्ट एग्जाम्पल है. The condition is the cells are locally situated. They are not closed. अगर आप ये वाला अगर देखोगे स्लाइड, if you see this slide, यहाँ पर ये पूरा का पूरा क्लोज है, राइट? दे आर वेरी टाइटली, दे आर hugging each other, but यहाँ पे क्या है? There is a gap. So paracrine में क्या होता है? Local signaling है, local signaling होता है. Okay, this is very important. This is called as paracrine, local signaling. Fine. एंडोक्राइन सिग्नलिंग. एंडोक्राइन सिग्नलिंग का कंडीशन सिर्फ और सिर्फ एक ही है. द कंडीशन इज वेरी वेरी फार. इट इज वेरी वेरी फार, वेरी वेरी फार. And how the signal is transferring? See, what is the medium? What is the medium? The blood. This cell will transfer its signal into the blood, and this blood will be going to the target cell, and here there will be the receptors, and the receptors पे ये सारा का सारा receptors जो signal molecules है वो bind होगा, and यहाँ पर signal transduction शुरू होगा. So this is called as endocrine signaling. और इसका सबसे बेस्ट एग्जाम्पल है हॉर्मोन्स. ओके, द बेस्ट एग्जाम्पल्स आर हॉर्मोन्स. सो दिस इज कॉल्ड एज अ एंडोक्राइन सिग्नलिंग. एंडोक्राइन सिग्नलिंग मींस द सिग्नल्स आर पासिंग थ्रू द ब्लड स्ट्रीम एंड द कंडीशन इज इट इज वेरी फार. ओके, the last one is the autocrine signaling. The last one is autocrine. Auto means apne aap, on its own. So see, the molecule is activating on its own. It is making the signaling molecule, then the signal molecule is coming out, and here is the receptor which is present, and it is activating itself. Okay, it is activating itself. This is called as autocrine signaling. This is called as autocrine. And one very good example is T cell activation. The T cells are activating themselves with the help of this autocrine signaling. Autocrine signaling is a very good example for this T cell activation. Whenever there is an infection, this T cell has to be in more numbers. So what it will do? It will automatically start its multiplication through this mechanism, autocrine signaling. Got everyone? Understood? Yes. Okay. I thought that I'm not audible. Fine. Okay. Now all these things I have explained you previously also. You can go through the slides. we will just, I have explained everything here. Modulator protein, anchoring protein. This anchoring protein is kind of scaffold protein only. We will do these questions. Now everyone will comment. Okay, if you have understood the things, you will very easily give this answer. The first question is, paracrine signaling is involved in which of the following? Please select all that apply. Hormonal communication, chemical signaling that can only travel limited distances, auto stimulation, synaptic transmission. D, synaptic, D. Now here, one thing, yes. Okay, but one thing you have to see here. See, I have written one thing here. Please select all, all that apply. So there is one more option. Yes, you are right. It is synaptic transmission. Very good. It is synaptic transmission. But here, one more option is there. Which one is that? Yes, very good, Deepshika. It is this. Because what is the term here? Limited distance. Limited distance. Yes, Millie. Limited means local. Limited means local. So paracrine signaling can be done both here. Chemical signals and synaptic transmission. Fine. Everyone understood? Now second question. Which of the following signal molecule is not used for extracellular signaling? Not. In your net exams, everywhere this type of questions comes. Not used, not has. So you should very carefully see this word. Yes, right. This is autocrine. This is extracellular signaling. Okay, extracellular signaling. A. Autocrine. Okay. Now see, autocrine, what I told, autocrine, this is the cell. यहाँ पे उसका सिग्नल मॉलिक्यूल है. ये सिग्नल मॉलिक्यूल बाहर आएगा. यहाँ पर एक रिसेप्टर प्रेजेंट होता है और ये सिग्नल मॉलिक्यूल यहाँ पे जाके अटैच करेगा. ओके, सो दिस इज अ काइंड ऑफ एक्स्ट्रासेल्युलर. एक्स्ट्रासेल्युलर मतलब क्या? ये बाहर आ रहा है. इट इज कमिंग आउटसाइड. इट इज एक्स्ट्रा. सो दिस इज नॉट द आंसर. ऑटोक्राइन नहीं है. एंडोक्राइन तो बिल्कुल नहीं है. पैराक्राइन तो बिल्कुल नहीं है. इट इज साइक्लिक एएमपी. ना ये जो क्वेश्चन है ना, इसको आप नेगेशन से भी सॉल्व कर सकते हो. सी, साइक्लिक एएमपी आई टोल्ड कि ये सेकेंड मैसेंजर है. सेकेंड मैसेंजर्स जो होते हैं ना, वो हमेशा सेल के अंदर होते हैं. दीज आर ऑलवेज इनसाइड द सेल. ये कभी भी सेल के बाहर नहीं होते हैं. दे आर ऑलवेज इनसाइड द सेल. Understood? This cyclic AMP, it is a kind of second messenger. So it is always inside the cell. तो यहाँ पे क्या क्वेश्चन पूछा था? It is an extracellular signaling. So now what is the answer? Yes, now what is the answer? Tell me. Yes, it is D. Understood? It is cyclic AMP. Fine. Next question. In endocrine signaling, एंडोक्राइन पता नहीं क्या होता है? ब्लड स्ट्रीम से पास होगा. आपका जो सिग्नल है ब्लड स्ट्रीम से पास होगा. द सिग्नल मॉलिक्यूल एक्ट ऑन द टारगेट सेल ओनली इन क्लोज प्रॉक्सिमिटी. ट्रू और फॉल्स? ओनली इन क्लोज प्रॉक्सिमिटी मतलब कि वो बहुत नजदीक होगा तो ही करेगा. फॉल्स. यस, राइट. इट इज फॉल्स. Fine. Now mark the signal molecule which does not interact with the cell surface receptor. Okay, fine. I have not told you this, but let's see. I'll explain you here. This question, it is like 10 times it has come in net exam, and during my JRF also this question has come in the net exam. Tell me. Does not interact with the cell surface receptor. Mark the signal molecule which does not interact with the cell surface receptor. Very good, Jancy. It is testosterone. No, Charmi, I'll tell you why is the answer. Whenever this type of questions comes, now does not interact with the cell surface, the answer is your steroid hormones. The answer is your steroid hormones. Okay. Only one thing you have to search in your options: steroid hormones, or whatever is relating to your sex hormones. This is a sex hormone. And what is the reason? Because these these steroid hormones can easily diffuse through the cell surfaces, so they do not want this cell surface receptors. इनको cell surface receptors की जरूरत ही नहीं है. They do not need this. They will directly enter into the cell. This question is very important. The answer is testosterone. यहाँ पे तो मैंने testosterone दिया था आपको ऑप्शन में, but whenever there is a question where it is written that it is directly passing the membrane, it is not interacting with the cell surface receptor, it is directly bounding the intracellular membrane proteins, the thing which you have to find is your steroid hormones. Steroid proteins you have to search for. Testosterone, estrogen, estradiol, like this, or steroid proteins, or directly यहाँ पे steroid दिया होगा. Understood everyone? The answer is testosterone. बाकी सारा कुछ भी options को देखने की जरूरत ही नहीं है, confuse ही नहीं होना है. सिर्फ एक ही चीज आपको याद रखना है, it has to be steroid. वही एक चीज है जो कि directly pass हो सकता है. बाकी सबको receptors चाहिए. Everything they need a receptor except this testosterone. And yes, small molecules भी pass हो सकता है, but typically for this type of question it is steroids. Understood? Name the signaling which requires physical contact. Very easy. Tell me. Physical contact. पैराक्राइन समझ में आ गया, इंट्रासेल्युलर समझ में आ गया. ए है मतलब इट इज अ काइंड ऑफ ऑटोक्राइन की बात करें, ये दोनों सेम ही है. Yes, it is juxtacrine signaling. It is juxtacrine. Juxtacrine में एक सेल दूसरे सेल के साथ फिजिकली कांटेक्ट में है और यहाँ से गैप जंक्शन से the molecule is passing. Okay. So I think everyone is understanding the steps. I hope so. Okay. Name the largest family of the cell surface receptor protein. Name the largest family of the cell surface receptor protein. Yes, very good. It is GPCR because it is a 7 trans membrane spanning protein and it is a GPCR. Next question. Neurotransmitters are released into the synapses at which of the following? Synapses मतलब कि the space. ये एक है और ये दूसरा nerve है. तो ये जो बीच का जो space होता है ना, इसको synapse कहते हैं. Now tell me, neurotransmitter are released into the synapse at which of the following? Pre है कि post है? Yes, it is post synaptic membrane क्योंकि pre इसको रिलीज करेगा किसके तरफ? पोस्ट की तरफ रिलीज करेगा. और पोस्ट पे क्या प्रेजेंट है? रिसेप्टर मेम्ब्रेन, रिसेप्टर्स प्रेजेंट है और ये रिसेप्टर्स में आपका क्या अटैच होगा? यहाँ से जो भी सिनेप्टिक मॉलिक्यूल्स आए वो यहाँ पे अटैच होगा और यहाँ पे पूरा का पूरा सिग्नलिंग सिस्टम जो है स्टार्ट होगा. सो इट इज पोस्ट सिनेप्टिक मेम्ब्रेन. Which of the following signal molecule is not used? अच्छा ये रिपीट हो गया क्या? Again, okay, tell me now. Not used for the extracellular signaling. Not used. D. Understood now? Yes, it is D. Yes, yes, correct. It is D. Okay. This is the last two questions. Arrange the following sequence. I was telling you now, this type of questions come. Arrange the following sequence of extracellular signaling in the correct order. You have to arrange this one. Transport of the signal to the target, start of the signal transduction, signaling cell synthesis, binding of the signal to the specific receptor. वेरी गुड. आपको यहाँ पे कुछ नहीं करना था, आपको सिर्फ फर्स्ट याद रखना है. अगर आपको पता चल गया कि पहले सिंथेसाइज होता है और फिर वो रिलीज करता है, दैट मीन्स यहाँ पे सिर्फ एक ही. अगर आपको बाकी का ऑप्शन नहीं भी पता है, आप आंख बिना देखे दूसरों को ये टारगेट कर सकते हो. इट इज 3142. खत्म बात. आपको बाकी के ऑप्शंस पढ़ने की भी जरूरत नहीं है, बाकी के ऑप्शंस देखने की भी जरूरत नहीं है. इट इज 3142 क्योंकि पहले वो सिंथेसाइज होगा, फिर रिलीज होगा, फिर वो क्या होगा? ट्रांसपोर्ट होगा, फिर वो बाइंड करेगा और फिर वो सिग्नल ट्रांसडक्शन पाथवे शुरू करेगा, राइट? या तो आपको फर्स्ट पता है या तो आपको लास्ट पता है, बट अगर आप लास्ट देखोगे तो यहाँ पे 2 है, तो ऐसे में फिर आपको थोड़ा सा एनालाइज करना पड़ेगा. सो इट इज, आप किसी भी तरीके से कर सकते हो. एग्जाम में क्या चीज क्लिक होती है वो डिपेंड करता है उस पॉइंट पे आप किस तरीके से क्वेश्चंस को पढ़ रहे हो. फाइन. सो दिस इज 3. फाइन. Which of the following signaling pathways followed by T lymphocytes in response to antigenic stimulation? Yes, yes, it is autocrine signaling. It is autocrine signaling. So I hope you have understood today's lecture, and I hope you have got to understand what is cell signaling, and I hope you have liked the lecture. So if you have any doubts or anything or any comments or any suggestion, you can tell me if you have any doubts. Everyone has understood the lecture. Okay, okay, thank you, Mansvi. Okay, okay, Jansi. Okay, Khushbu. Okay, Millie. Okay, Priyama. नो डाउट्स ना, मतलब या तो कुछ नहीं समझ में आया या तो सब कुछ समझ आया. आई विल गो विद द सेकंड ऑप्शन, सब कुछ समझ में आया है. सो एनी टाइम एनी क्वेश्चन इज आस्क्ड फ्रॉम एटलीस्ट दिस पार्ट, you can be easily able to solve those questions. From here, each and every point was important. I cannot I cannot define that okay ye portion bahut important hai, ye portion bahut important hai. Everything was important. And from here B में तो questions आती है part B में, and part C also there are questions जो कि मैंने आपको वो कैंसर वाला जो टारगेट वाला बताया था उस टाइप्स में questions आते ही हैं part C में. And that is very confusing in the question, but the answers बहुत ही simple होता है. नेक्स्ट टाइम व्हेन वी आर डूइंग इट इन द स्ट्रक्चर्ड क्लासेस मैं आपको सी टाइप के भी क्वेश्चंस बताऊंगी कि वहां पे कैसे आते हैं. बट ये सारा का सारा जो था दिस वाज टारगेटिंग द बी पार्ट में आपको ऐसे ही क्वेश्चंस मिलेंगे. फाइन, फाइन.
Okay, ma'am. Okay, ma'am. So, students, as you all can see, how these classes and the tests are going on. Ma'am and sir is explaining everything. Because
Okay.
CSIR NET has very vast syllabus and all, so we cannot discuss all those things in the demo classes. हम लोग हर चीज डेमो क्लास में डिस्कस नहीं कर सकते, तो आप लोगों को प्रीमियम लेना बहुत ही जरूरी है. आप बस अपना एम रखें कि CSIR नेट को क्रैक करना है, और हमारे जितने भी फैकल्टीज उनमें से ऑलमोस्ट सारे ने CSIR नेट क्वालिफाई किया है, और मैम ने खुद स्ट्रेटेजी और एनालिसिस आपको फर्स्ट लेक्चर में बताया है. तो आप जितना जल्दी रजिस्टर करेंगे उतना जल्दी हम लोग मेन स्ट्रीम क्लास, वो स्ट्रक्चर क्लास जो है वो स्टार्ट कर पाएंगे. तो आप लोग से गुजारिश है कि आप लोग जल्द से जल्द रजिस्टर करें, और रजिस्टर करने के बाद ही आपको क्वेश्चन पेपर्स या फिर PDF जो है, जो पावर पॉइंट्स आपको मिल पाएंगे. क्योंकि सिर्फ लाइव क्लास पढ़ने से नहीं, उसका नोट्स बनाने से भी होता है. आपको तभी अच्छे से समझ में आएगी जब एग्जाम टाइम आएगा तो आप नोट्स को प्रेफर करोगे. तो आपको PDF पावर पॉइंट मिलेंगे तभी तो आप नोट्स बना पाओगे. तो आप लोग से यही गुजारिश है कि जल्द से जल्द रजिस्टर कर लें. ऑलरेडी कुछ बच्चों ने रजिस्टर कर दिया है, तो आप लोग भी जल्द से जल्द रजिस्टर कर लें. जैसे कि आपने यहाँ पे देखिए प्रियमा कुमारी, उसने ऑलरेडी रजिस्टर कर दिया है, तो आप लोग भी जल्द से जल्द रजिस्टर कर लें. और आज के सेशन के रिगार्डिंग कोई डाउट हो तो अभी आप पूछ सकते हैं. इफ यू हैव एनी डाउट्स रिगार्डिंग टुडेज सेशन you can ask. And again I want to repeat, first 30 comers will have concession of 500 rupees, and you can pay in the installment also. No need to worry about, just discuss with your parents. Already few students are discussing with their parents, and don't worry, we will work hard with you. Ma'am has already shared the strategy analysis.
If you have any suggestions that what we can do or what